We don't do single viruses in this course because it's meant to teach you principles of virology, and I think the way you learn that is not to go over individual viruses. But today is an exception. We're going to talk about HIV and AIDS because this is clearly the most important viral pandemic of the 20th century. If you're interested in the origin of AIDS, which we'll talk a little bit about today, this is a wonderful book written by a physician who worked for many years in Africa and got together a lot of information about how he thought HIV might have spread in the early days. And I've borrowed quite a bit from him today. And this is a perfect quote from his book. This tragedy, referring to the AIDS pandemic, was facilitated or even caused by human interventions, colonization, urbanization, and probably well-intentioned public health campaigns. And if you read this book, you'll see that it would have been hard to prevent this pandemic, but we can clearly see some of its origins. So the AIDS pandemic begins with this article in Morbidity and Mortality Weekly Report, June 1981, a report of 5 young men, all active homosexuals, treated for biopsy-confirmed pneumocystis carinii pneumonia at 3 different hospitals in Los Angeles. 2 of the patients died. All 5 had laboratory-confirmed previous or current cytomegalovirus infection and candida. And then this is a case report of these cases. And this was a very wise observation. These were the first physicians to note that something unusual was going on here, which had otherwise been ignored. Pneumocystis pneumonia in the U.S. is almost exclusively limited to severely immunosuppressed patients. So the docs who wrote this were surprised that there were suddenly 5 cases in a cluster of this very rare disease. All of the above observations suggest the possibility of a cellular immune dysfunction related to a common exposure that predisposes individuals to opportunistic infection. So that was the beginning of AIDS. It took many years, though, before it was recognized and even acknowledged. The president of the U.S. took years to even mention the word AIDS. Now, AIDS was further observed after that first observation by clusters of pneumocystis pneumonia and Kaposi's sarcoma, which is another rare disease, cancer, in other urban centers. So these began to crop up all over the U.S. And the CDC established a case definition. So they said, here's what you should look for. There's an unusual disease. Look for Kaposi's sarcoma or opportunistic infections, a rather straightforward case definition. 1982, about a year later, the disease was renamed AIDS, acquired immune deficiency syndrome. It was first called gay-related immunodeficiency disease. And this idea that it was a strictly gay-related disease persisted for so many years. And Reagan didn't acknowledge it until his buddy Rock Hudson died, right, of AIDS. And it took that for Reagan to say, we have a disease called AIDS in this country. This is absolutely unacceptable. It was subsequently found to be transmitted both at birth and heterosexually, in addition to homosexual transmission. And the virus turns out to be a lentivirus, which is a member of the retrovirus family, first isolated in 1983 from a lymph node of a patient with swollen lymph nodes, lymphadenopathy in Paris. There's a big controversy over whether Robert Gallo in the U.S. identified it, but all he did was get cells from the French and pull out their virus. The first discovery clearly goes to the French, and they were awarded the Nobel Prize for this many years later. A blood test was developed in 1984. Once you have the virus, you can develop a blood test and start screening not only people for infection, but the blood supply. For years, this was being passed in the blood in the U.S. and passed on to many, many people. Electron microscopy and sequence analysis of the genome revealed that the virus was a lentivirus, which is a known group of retroviruses. So we've talked a lot about retroviruses in this course, and we haven't discussed the various classifications of them, but now you can put it all into perspective. There is a subfamily of the retroviridae called the orthoretroviridae, which contains most of the retroviruses we've talked about. Avian leukosis virus, the progenitor to Rous sarcoma virus, is an alpha retrovirus. Human T-cell lymphotropic viruses, 1, 2, 3, very interesting viruses that cause cancers in people. Those are delta retroviruses. We mentioned them very briefly. They're examples of viruses that take a long time to transform cells. Epsilon retroviruses, we mentioned one example, the walleye dermal sarcoma virus. And then there are the lentiviruses, which contain HIV 1 and 2. There are 2 types of HIV. There are really 2 distinct groups of human retroviruses that infect humans. There are retroviruses that infect many, many other species on Earth. We have the lymphotropic viruses, HTLVs. There are 4 serotypes of those, and then the immunodeficiency viruses. Now, when HIV was discovered, it was clear that it wasn't anything new. It was new to people, of course, but there are other lentiviruses that have been discovered before. One causes a fatal disease of horses, isolated in the early 1900s. And the equine infectious anemia virus, as it's called, causes an immunosuppression very much like HIV does in people. So HIV is a retrovirus. You've seen these before. These are enveloped viruses. They have a glycoprotein in the membrane. Inside the envelope is a capsid that encases the plus-stranded RNA. Now, this capsid is a slightly different shape from some of the other retroviruses we've talked about. You can see the 2 copies of the RNA, and not shown here, of course, are all the viral proteins that are in the capsid, including the reverse transcriptase, etc. The genome of the virus is shown at the bottom. This is the proviral view. Remember, the provirus is the DNA integrated into the host cell. So we have the 2 LTRs at either end, and we have the typical GAG, Pol, and envelope genes, which we've seen in the other retroviruses that we've discussed. Now, the retroviruses we've talked about up until this point, we call retroviruses with simple genomes. They encode 3 precursors, GAG, Pol, and envelope. All the viral proteins are derived from them. The structural proteins from GAG, the enzymes from the Pol region, the glycoprotein from the envelope. But HIV is a member of retroviruses where we say the genome is complicated, or retroviruses with complex genomes, because they encode a lot of other proteins, which you can see here. VIF, TAT, REV, VPR, VPU, TAT, REV, and NEF. And all these proteins have very specific functions. We've talked about some of them in terms of immune antagonism, in terms of getting RNA out of the nucleus if it's not completely spliced. And so these are unique to these lentiviruses, having all of these extra, we call them accessory proteins, but they're very important for replication and pathogenesis. AIDS, of course, stands for acquired immunodeficiency syndrome. And again, the virus was renamed about a year after its discovery. The syndrome, of course, is the occurrence of a characteristic group or pattern of symptoms. Often you call a disease a syndrome before you figured out what causes it. Now we know, of course, that HIV causes AIDS, but the name has stuck. And HIV is the agent of epidemic AIDS. There are many AIDS denialists in the world. All right, I encounter them often, and they tell me that there's no proof that HIV causes AIDS. This, of course, is nonsense. Many times people have stuck themselves with a needle in a hospital, a contaminated needle, and have gotten AIDS. I don't know what a better proof of the virus causing AIDS than that, but it's happened over and over. And as we see, we can treat people with antivirals, and they get better. So HIV causes AIDS. Don't ever think or don't ever doubt that it does, because there's absolutely 1,000% proof that it does. Let's talk a little bit about the extent of the epidemic first. In the United States, deaths from HIV over 600,000 since the beginning, and this exceeds all U.S. combat-related deaths in all the wars that we have fought in the 20th century. You know, we fought a couple of big ones, and a lot of people died in those, but AIDS has killed more. At the moment, we have over 1.2 million in the U.S. infected. 13% of them are unaware that they are infected. As you'll see, they haven't yet gotten into the symptomatic phase. This is just in the U.S. now, 50,000 new infections a year, 70% in men, 30% in women. Half of all the new infections in the U.S. occur in people 25 years or younger. And these are the different ethnic groups that are getting infected on the x-axis, or the number of infections every year in the US, and you can see white men who have sex with men, black men who have sex with men, etc., Hispanic Latinos, black heterosexual women, black heterosexual men, white heterosexual women, etc. So there are different risk groups. Now, why is this? Since we know HIV causes AIDS, we know how to prevent it by engaging in proper protection during sex, yet people feel, well, we have antiretrovirals now, it doesn't matter, and they engage in risky behavior. This is a completely preventable disease, at least in the US. Let's take a look at some of the numbers globally, because this is really quite stunning. These are from UN AIDS. This is up to 2014, so they lag a little behind in the numbers. Number of people living with HIV, 36.9 million globally, and that's the range, because this is an estimate, obviously, the number of adults and women. And this is the real tragedy. Children less than 15 years of age, they get it typically at birth. They are not even involved in acquiring the virus. They get it from their mothers who are infected at birth. People newly infected in 2014, 2 million, and broken down as well. And then the AIDS death, 1.2 million, broken down as well. If you look at the adults and children estimated living with HIV on a global perspective, you can see sub-Saharan Africa has the burden, the vast burden of the disease. And this is in part, as you'll see, because it originated there and had a lot of time to spread before we recognized it, and because the drugs that we use elsewhere aren't fully deployed in Africa. But other parts of the world have many millions of cases as well. So this is adults and children. Newly infected in 2014, again, you can see the vast majority in sub-Saharan Africa. But still, these numbers are very high, 2 million newly infected adults a year. Estimated adult and child deaths, again, the most in sub-Saharan Africa. And in many places in Africa, this is wiping out local populations. Children estimated to be living with HIV, 2.6 million globally, most of them in sub-Saharan Africa. Here, for example, in the US, we know when a pregnant woman is infected, we can diagnose her early on, and just before birth, we can give her a dose of antiretrovirals that will vastly reduce transmission to the baby at birth. This is the number of children newly infected in 2014, 220,000. Again, most of them in sub-Saharan Africa. And finally, deaths in children less than 15 years of age. This is a tragedy, again, because they had no say. If you want to engage in risky behavior, that's one thing, but kids don't have any say in getting infected. There are about 5,600 new infections a day in 2014, 665 in sub-Saharan Africa, 600 in children, and 5,000 in adults, half of them in women, and 30% among young people. So this is still a serious problem, obviously. So here's a graph showing you the new infections and deaths from about 1990 to 2011. It's always hard to find graphs of the newest data, but this is a trend that's continuing. You can see the new infections peaked around the late 90s, and then public education, telling people how to stop spread, coupled with antiretrovirals, has brought down the number of new infections. The number of deaths lagged slightly behind. They peaked in about 2006, 2007, and they are starting to decline as well. So we're making some inroads at stopping infection, but it's going far too slowly. Now, one of the key tools, of course, is triple therapy, which we talked about when we talked about antivirals. There's a single pill. There are a couple of them. This one's called Atripla. That will reduce the likelihood that you will get drug resistance, which has to be taken every day, unfortunately. And this has certainly slowed the pandemic. And if in countries with money, of course, it slowed them a lot faster than in other countries. So here's a graph of the numbers of people receiving antiretroviral therapy globally. And this is divided into the different WHO regions. You can see the African region, Americas, Southeast Asian, European, et cetera. And you can see the people receiving antiretroviral therapy has gone up substantially since 2000 up until the present. The vast majority, again, in the African region, which is where it's needed the most, but in other countries as well. But the bottom graph shows you the global antiretroviral coverage in percent. This is the percent of people who need it. So a percentage of the people who are infected. So you can see in 2014, we're only hitting about 40% of the 39 million or so people who are globally infected. So we have a ways to go. We're getting better, but we need to get these drugs to everybody. The problem, of course, that even though the drugs can stem infection, you have virus in you for the rest of your life. There's no cure. You cannot clear virus from an infected individual. There's no vaccine, so we can't block primary infection. Talk a little bit about vaccines at the end. The future is not looking very good in terms of the trials, but there are other promising approaches. You have to take your antiviral drugs every day for the rest of your life. Now, there are many people who can live very good lives doing this, but if you stop for a few days or a week, the viruses that are in you, your proviruses that exist in very long-lived hematopoietic precursor cells, they will start to make virus and you will become infected again. Of course, if you stop taking your drugs, you will get drug resistance as well. The drugs are expensive. All of this means that it is a third world disease, pretty much spreading very swiftly in sub-Saharan Africa. So we have a lot of work to do. It's not over by any means. This is a disease that can be stopped, but we have to get the drugs to the right people. Now let's talk about where HIV came from. It clearly originated in Africa. There's no doubt about it. Now remember, it was discovered in 1982, and so immediately, once a blood test was developed, people started looking throughout the world in different populations by serological surveys and saying who's infected. And look at this. The first studies in Africa, in Zaire and Rwanda, showed that AIDS was common in the capitals of those countries, Kinshasa and Kigali, the capital of Rwanda. 90% of sex workers are infected. This is way at the beginning of our discovery, 1982, 1983. Already 90% obviously had been in this country for a very, very long time. And the fact that so many sex workers were infected gave a clue as to how it was predominantly spread. People began testing frozen, stored samples in various places, and they found clearly evidence for infection in the 60s and 70s in locations in Central Africa, but not in West or East Africa. So here's the belt right around here where we're seeing most of the HIV infections. A sample from a male in the Democratic Republic of the Congo that had been stored, this famous serum sample called ZR59, was shown to be positive for HIV in 1998. So that's the earliest confirmed infection by serological means, and then subsequently a lymph node sample from a woman in the DRC, which was obtained in 1960, was also shown to be HIV positive. So those were important samples because they differed by about 12%. They're a year apart, 1959, 1960. The sequence of the genomes, they eventually recovered genome from these samples as well, were 12% different. So that gave us a clock on how fast the virus was changing, and we could make the clock calculation based on 1959 to the present. And so we could say for sure, not only was the virus present in Kinshasa, which is here, but it was also there in the 50s and 60s, and it also enabled backdating of the origin, which looked like the first part of the 20th century. Now we have to talk a little bit about the way HIV is classified. There's a lot of diversity of HIV1. It's classified into 4 groups entirely based on sequence alignments. So these groups are called M, N, O, and P. And M stands for main because it encompasses 99% of all HIV-1 infections. But there's another group called O, which stands for outlier. It has nothing to do with the alphabet. We didn't go from A to M. We started at M. O is outlier, less than 1% infections, and limited to Cameroon, Gabon, and some neighboring countries. There are only 13 cases of group N, HIV-1 in Cameroon, and 2 cases of group P in Cameroon. So you see, group N is the main one that we are worried about. But it's interesting, each of these 4 groups originated from a separate introduction of virus from an animal into a person. So group M was one introduction, group N, group O, and group P. Separate introductions. 4 different crossovers. Now, the groups are further, for group M, it's so extensive in its infection that it is further diversified into subtypes, 9 different subtypes. Furthermore, people who are at high risk for multiple infections, so sex workers will get multiply infected, right? They will get one strain from one client and another strain from another client. They can be infected many times, and these viruses multiply in them, and they recombine their genomes. So you get recombinants, and some of these recombinants are very fit, and they predominate and spread through the population. We call them circulating recombinant forms, and there have been 48 of those detected so far. We detect them because they spread. There are probably many, many that develop but don't get anywhere. Now, you may be wondering if these subtypes have any difference in their ability to spread or cause disease, and there isn't any evidence except that the D subtype, we seem to think they die a little faster, but it's not quite clear why. Also, the shedding of subtype C in the female genital tract is higher than the others, and this may result in higher female to male transmission and could partly explain the extensive spread in Africa. So basically, when an individual is infected with HIV, it evolves in one direction. So we can always tell who has infected a person with what virus, because the virus evolves in one direction from a single input virus. And we think people are infected with very few viruses, and it evolves to subtypes and recombinants. So if you look at the sequence of HIV isolates in a particular country or region, you can look at how infection has progressed. In fact, there have been some very well-publicized court cases where people have sued other people for giving them HIV. There's a famous case where a guy, infected 1,000 women with HIV. He found he was AIDS positive and wanted revenge. I don't know how that got him revenge, but a number of these women sued him, and they did genome sampling from all the women and from him, and they could prove beyond a reasonable doubt that it was him who infected all of them. There's a very famous paper that was released with this trial. And this is all because we can do nucleotide sequencing of large numbers of isolates. We couldn't do that before. That technology arose in the 90s. Now, all of this sequencing revealed to us that there's more diversity in Central Africa than anywhere else. So if the virus evolves in one direction, from a virus that infects you to within you to the next person and so forth, that means that HIV has had more time to evolve in Africa than anywhere else. And that's another reason why it's quite clear that Africa was the origin of HIV and AIDS. Now, some subtypes are associated in specific regions with particular modes of transmission. And this is because of what we call the founder effect. In a particular risk group, in this case, white homosexuals in South Africa, that's a very specific risk group. If you introduce a virus into them, they tend to spread it amongst themselves. And so that subtype will predominate in that particular group, in that particular area. So, for example, subtype B is found in 96% of white homosexuals in South Africa, and it came there from the US. So it was introduced to South Africa from the US, and then it spread specifically within that risk group. And type C accounts for 81% of infections of black heterosexuals. So you can get an idea of the groups that are spreading these viruses because these subtypes are so specific in this way. So here's a global map of subtype distribution. All right, so those are all the 9 subtypes and the circulating recombinant forms derived from group M, and each of the pie charts is color-coded. So, for example, in the US, we have mostly subtype B and a very little of subtype C. And again, this is all because of what initially spread somewhere else. So the preponderance of B in the US is because the founder strain was a B. It entered early and had time to spread before other strains could come in as well. So subtype C, B, and A, and G, et cetera, this are the percentages. You can see subtype C is a big one, but all the others are present as well. And F, H, J, and K have limited transmission. Now you can see, for example, in Central Africa, which is expanded here, pie charts have lots of colors. again, it's the most diversified area. The virus has had time to diversify the most in this area. And you can see in South Africa, we have a lot of subtype C and so forth. And this is the global distribution. Subtype C is, again, predominant. That's largely because of Africa. Now, what was the source of HIV? This is a long work in progress that's been done by a number of groups, but the main driver has been this individual here, Beatrice Hahn at the University of Pennsylvania. And that's her picture in at the wall of polio when it used to be in my lab. It fell down so many times that we moved it to my office. Beatrice's daughter took this course one year, all right? And she didn't tell me she actually took the course until the next year when she wanted to TA. She said, I'm Beatrice Hahn's daughter, and I want to TA your course. How about that, right? Isn't that cool? And she features predominantly in this story. So she got together an army of people to look for the origins of HIV in Africa. I heard her give a seminar many times, but she says she has an army of shit collectors, because that's what they do. They go collecting chimpanzee shit, because it turns out, we thought for many reasons that the origin of HIV was in chimpanzees in a virus, a simian immunodeficiency virus, which was first isolated from chimps in 1989, and it turned out to be very similar to HIV. And it's very hard to capture chimps and bleed them and let them go. They found out that if you collect shit, that you can find virus in it. And you just need to, they live up in the trees, and then you can collect the feces from the grounds. You can even get virus from urine. And then these guys would hold collections. In the morning, apparently the chimps wake up and urinate over the side of their tree home, and these collectors would stand there and try and collect. And then they could do mitochondrial DNA typing and identify which chimpanzee it is. So all of these dots here are individual sampling sites for chimpanzees in this belt of Central Africa. And there are many different, a number of different kinds of chimps, different subspecies. Probably the ones that you know the best are Pan-troglodytes, but there are Verus elioti, Schweinfurthi, and then there's the bonobos, Pan paniscus. And not all of these chimp colonies were infected. She looked at over 7,000 fecal samples from 90 sites, and not all of them have SIV-CPZ, but a good number of them. Now, chimps form these independent colonies. They don't interact with each other, so the infections, you can have one that's infected, which is colored, and others which are not infected at all. And especially if there's a river between the colonies, the chimps never swim, so they will never cross the river. Anyway, she defined very close relationships among the chimps, and she found that the chimps from Cameroon were actually the closest to our contemporary HIV. Now, SIV is transmitted among chimps sexually, but also by birth, and chimps fight, and they could probably transmit it by blood then. And the transmission frequency per coital act is this number here, which is similar to that is human. And the virus is pathogenic in its natural host, chimps. It causes AIDS. It causes an immunosuppression. And many chimps have died over the years of SIV. And from Beatrice's studies, well, first of all, let me go back 2 slides. It turns out that Pan-troglodytes was the main reservoir of the SIV that was closest to HIV. And the other species did not harbor that, and that was from Cameroon. It turns out now that after many years of work, this is what we understand about the origin of the HIV groups. Remember, there are M, N, and P and O groups, 4 separate transmissions from monkeys to humans. They all begin with SIV viruses in different monkey species. So when we move away from the great apes, the chimps, and the gorillas, we move back a little in evolution, we encounter the monkeys. So all these monkeys here in this circle, mandrills, mangabes, vervets, etc., each of them has their own SIV. We know this from sampling. And in those monkeys, there's no pathogenesis from being infected. Now, HIV-2 is the result of a transmission of SIV from a sooty mangabe to people. We'll talk a little bit about that in a minute. Now, the HIV M and N group, so M is the biggest group in people, that came from two transmissions from a chimp SIV, so SIV chimpanzee, but the chimp was relatively recently infected. The chimps have not been infected that long. They acquired their viruses from Mona monkeys and red-capped mangabes. In fact, these two viruses that circulate in chimps, the virus, the SIV-CPZ that circulates in chimps is a recombinant of a Mona monkey and a red-capped mangabe SIV. Now, because it's a recent introduction into chimps, it's pathogenic in those animals. It hasn't had a long time to evolve. And chimps, on two separate occasions, have passed SIV-CPZ to humans, the M and the N. And this has been from pan-troglodytes troglodytes. It's quite clear that it is that species and not the other ones that harbor the origin or the ancestor of HIV in people. So how did chimps get it from monkeys? They probably ate them. We think that chimps are vegetarians, but it's not true. They will eat meat and they will fight with other monkeys on occasion, not too frequently, and that's probably how they acquired the SIV from monkeys. Now the O and the P groups we obtained from gorillas. Of course, you're probably thinking, how do we get it from chimps and how do we get it from gorillas? Because we don't often fight with them, but we'll get into that in a minute. The gorilla, in turn, acquired it from a chimp. So you can see here, the SIV-CPZ, which has gone twice into people, went from a chimp to a gorilla, and then from a gorilla to a human. How did the gorilla get it from the chimp? Probably fought. Again, they don't do that very often, but they could fight and exchange blood and get infections. So this is the result of many, many years of sequencing and ecological studies, and it's now quite clear where we got these viruses from. Now, when is a different story. Now, to do that, we need to look a little bit into sequencing. And a few times in this course, I've shown you some phylogenetic trees. So I think I'm going to show you one again. I want to make sure you understand what they are. Phylogenetic trees measure the genetic distance between organisms, and you can identify the nearest relatives of any given organism, and if it's a virus, you can do the same thing. And each division is a tree, is called a node, and then we have a presumed ancestor to each node. We don't often have the ancestor, because remember, a tree is made by sampling what we have now on Earth, the viruses that we now have. So we can infer what an ancestor of any 2 or 3 viruses is, but we don't have the ancestor for the most part, all right? So these are inferred. When you have an ancestor and it branches, say, into 2 viruses, they evolve independently. So the root at the left is a common ancestor that's assumed. And so the way we make these is we compare sequences of different isolates of the viruses, and we like to do it for individual genes. So the retroviruses have GAG, PAL, and OMV genes, and often they will compare the sequences of these separately to make phylogenetic trees. All right, so now that you understand what that is, here are 3 trees that show you basically what I've just told you. The origin of HIV lies within chimp and gorilla SIVs. So here on the left is a phylogenetic tree for the GAG gene only. And you can see in red are the HIVs, the human isolates. So we have M up there at the top, and we have N here. And we also have the P and the O isolate. So those are the 4 different groups of HIV. And you can see the O clusters with a gorilla isolate, gorilla SIV right there. So that is very close to these 2 O isolates. They have a common ancestor. And that's part of the reason why we think we got this virus from gorillas. You can see the same thing here with HIV-P, clusters with gorilla SIV. And then, of course, the N and the M HIVs cluster with the chimp isolates, SIV-CPZ from pan-troglodytes troglodytes. You can do the same thing with the PAL and the envelope and the NEF genes. You get the same result. So the more genes you do this with and confirm the result, the more confident you are that it's true. We know where HIV came from. Again, 4 different introductions from chimps or gorillas. How did it infect us? Main theory is called the cut hunter theory. At some point, a hunter, a man or a woman, we don't know who it might be, was hunting, and if it was quite a while ago, that was the main way of getting food. Maybe a hunter killed a chimp, and while dressing it in the field, could have gotten cut and gotten blood into his or herself. In that book by Jacques Pepin, which I told you about, he goes through a number of calculations. And you can calculate that in 1921, the number of people infected with SIV was less than 10. So probably as people hunted, they got multiple infections with SIV, CPZ, and very few of them ever went on to be a pandemic. So about 10, but only one probably spread. So think about this. There was one hunter hunting a chimp. He or she got cut, got virus into him, it replicated, he spread it, and now that's why we have an AIDS pandemic today, from one hunter. And this is the way emerging viruses work. And as I say, these infections probably have happened over and over. They're probably happening today because we know there's still bushmeat hunting going on. We get Ebola coming over from bushmeat. So there could be that SIVs are coming over as well today. But why did this one spread? You know, M, the majority of HIV infections, why did that particular crossover spread? M and O came across from an animal in the first 3 decades of the 20th century. We don't know when M and N and P came. Remember, M and O, M and N from chimps and O and P from gorillas. We don't have enough data to date those, but M and O from the first 3 decades. And it looks like Kinshasa was the epicenter of the outbreak, and it spread throughout Africa, correlated with the development of colonial cities. Now, if you know anything about the history of Africa, you know it existed on its own as an unexplored place with tribes and kings and so forth ruling the country. And then at one point Europe said, we belong in Africa and it's ours. And they decided to move in, and every country claimed a different part of Africa. This is what we call colonialization. And we think that had a great deal to do with the spread of HIV. Here on the upper left is a graph of the growth in population of various African cities from the late 1800s to 1970. You can see very, very high population growth in many of the larger cities in Africa, and many of these were established here. You can see the dates for establishment of Kinshasa and some of the other cities as well. Now, Leopoldville was the most dynamic city in the region. What was happening is as cities grew, people would come in from the surrounding woods to work. Okay, they would previously be working, capturing animals and so forth. They would become migrants and move into these big cities. So the theory is maybe the cut hunter moved into one of these cities. And as populations in cities grow, so do brothels pop up and people visit brothels. And he may have gone to the brothel and then a STD clinic when he came down with a sexually transmitted disease. So if he went to an STD clinic, now many of these European countries were establishing clinics in these growing African cities to keep the workforce healthy. But the problem is they used glass syringe needles and they didn't sterilize them properly. So if they treated him for his STD, they probably spread his infection to other people as well. And of course, he would have sex with other people. Some of the women in these early brothels would have 1,000 clients per year. Now, the Belgian Congo is an interesting story. This is part of the whole spread. The Belgian Congo was taken over initially by Belgium, of course. In the early 60s, the people who lived there revolted and told the Belgians to get the hell out. But then when the Belgians left, the local people found they had no doctors left, right? So the Haitians volunteered to come and take care of people in the Congo. There were physicians there. They spoke French, the same language. And they worked there for a number of years and mingled with the population. And when they went back to Haiti, they took AIDS with them. And Haiti was one of the early places of spread for HIV to the US. So this simple act of, insinuating into the population in the Congo helped spread the infection. Colonization, as I said, played a big role in establishment of large population centers, prostitution on a large scale, introduction of healthcare, as I've mentioned, colonial medicine, injections, transmission of viruses with unsterilized syringes. And I think I've mentioned this before, there are plenty of precedents for us spreading diseases using unsterilized or poorly sterilized equipment. In Egypt at the turn of the 20th century, they tried to get rid of schistosomiasis by injecting people with anti-parasite drugs. They spread HCV to over half of the Egyptian population, and we're still trying to deal with that today. So all of this probably resulted in large-scale amplification of HIV-1, and eventually it moved out of the country initially with the Haitians and then to other parts of the world as well. Now, HIV-2 is a very different virus, only 30 to 40% identical to HIV-1, first isolated in Guinea-Bissau, mostly in populations of West Africa, not very pathogenic. Most infections don't progress to AIDS. It's less transmissible. There's no mother-to-infant spread. And as I said, this is resulting from a crossover from an SIV and a sooty mangabe, which many people in this region keep as pets. They capture them and they keep them, they scratch you, and they infect you. There have been 8 separate introductions of SIV, HIV-2 from SIV and a sooty mangabe into people. So there are 8 distinct lineages. How is HIV transmitted? It's not a very infectious virus. It's not contagious like measles. The R0 is from 2 to 5. It is not spread by respiratory, eating, or vector routes. And here on the bottom are the ways that it is spread. It really differs according to the country or region. In the US, most of the spread, 62% men having sex with men. In Eastern Europe and Central Asia, mostly spread by intravenous drug injection, contaminated needles, spreading contaminated blood from one person to another. In Latin America, commercial sex workers and their clients. And in South and Southeast Asia, we have also commercial sex workers and their clients. Now, other ways of spread, so we have sex, we have contaminated needles, intravenous drug use. The blood supply used to spread it. We screen most blood now for HIV. And mother to child at birth accounts for about 5% of the spread. We don't actually think the virus crosses the placenta, but rather at birth, when there's a lot of blood around, the blood infects the baby. So if you can bring the retrovirus level down just before birth with drugs, that really knocks down the transmission to the baby. Now, if you look at virus in body fluids and cells, that's shown on this chart. So here in the center column, we have the number of virus isolations and the number of samples that were tested, and these are all cell-free viruses here, so you can see the quantity of virus here. A lot of virus in cerebrospinal fluid. There's a lot of virus in plasma, in semen. And then if you look at infected cells, PBMCs, and semen as well. We also think that vaginal cervical secretions are a large cause of transmission, although we don't have an estimate on the quantity of virus. We know that these are positive for virus. So we think that either virus or virus-infected cells can transmit infection, and the presence, the high levels in some of these sites suggest how transmission might occur. Now, a number of studies have been done to figure out what's the probability of acquiring HIV per sex act, okay? And this is an example of a study done in Uganda. They take couples, male, female, married couples, who are discordant for HIV. One is infected and the other is not. They ask over time, what's the fraction of the other person getting infected? And this is related to viral load. So the bars on the x-axis is the viral load of the infected partner from very low to very high. And then on the y is the probability of transmission per 10,000 coital acts. And so here you can see with low RNA it's very low, but if you have a high viral load, it can be up to 50 per 10,000. And if you have genital ulcer disease, if you have open sores, this improves the transmission. And that's what the red bars are. Blue is without genital ulcer disease, red is with. So that's 0.001 is about the risk of transmission per coital act among humans. So that's a low number, but it could occur the first time, in the middle, at the end. Here's another way of looking at the risk of transmission. So sexual transmission the different ways, female to male, male to female, male to male. You can see the numbers here. Parenteral, which means by a needle stick or blood, transfusion of infected blood is very efficient at transmitting infection. So early on in the epidemic, before we knew to test blood, it was being spread like crazy, especially among hemophiliacs, which needed regular infusion of red blood cells. Needle sharing 1 in 150, a needle stick 1 in 200. But if you get a needle stick in a hospital, say, and you know that the needle is from an AIDS-infected patient, if you then take antiviral drugs, in this case AZT, pre-exposure prophylaxis. So people who work in high-risk settings can take antivirals as a pre-exposure protection. You vastly reduce the risk of infection. But nowadays, if you get stuck with a needle and the needle has been contaminated, you immediately get an antiretroviral and you will not get established infection. Finally, mother to infant, 1 in 4, very high transmission without AZT. But again, if you treat the mother with AZT, you vastly reduce the frequency of transmission. Other factors to consider in transmission: the virus is inactivated readily by just drying on surfaces within a day. It's inactivated by heating, bleach, or alcohol, extremes of pH. So the virus is not very sturdy. And that is why it has evolved first in chimps and then in humans to be spread by intravenous drug use, sexually transmitted diseases, routes that bypass anything that would inactivate the virus. So that's what evolution does. It selects for viruses that are transmitted in certain ways, so the virus is not inactivated. The virus, as we mentioned a long time ago, binds to a CD4 receptor on the cell surface. So the CD4 is, as you know, present on T cells, CD4 positive T cells, and those are shown here. But the virus requires 2 different receptors to enter a cell. It needs CD4 and a co-receptor to get high affinity binding. And the co-receptor is a chemokine receptor, which can be one of 2 kinds, CXCR4 or CCR5. Now, it's very interesting that when you are infected, when you acquire HIV infection, it's usually a strain that binds CCR5. And then as the virus evolves in you, it's typically a CXCR4 binding strain. But if you transmit it to someone else, you invariably transmit viruses that bind CCR5. So there's some bottleneck with respect to the population as it controls receptor utilization. Very interesting. Let's talk about the pathogenesis of infection. Primary infection, virus crosses the mucosal barrier. One of the primary cells we think is the dendritic cell, to which the virus binds initially. And again, this occurs with CCR5 binding strains. Those are the ones that transmit. There's a protein on the surface of dendritic cells called DC sign. The virus seems to attach to it, but it doesn't get into the cell and replicate. And the DC does not get activated as it would normally by binding a foreign antigen. So the DC is not activated. And when the DC goes into the lymph node, it brings the virus with it. And in the lymph node, the virus comes off, and there are all the cells the virus wants to replicate in, CD4 positive T cells. So it's very interesting. This strategy of surveillance by the dendritic cell actually brings the virus exactly to where it needs to go to replicate. Virus replicates in lymphoid tissues. Lots of viruses released into the lymph and then into the blood. You have a lot of viremia. And then the virus goes to all the other parts of the body that have CD4 positive cells in it and replicates there. After a few weeks, you have an immune response to infection, and the virus load goes down to a certain set point. After about 6 months, you have that amount of low virus for many, many years until you develop disease. So you have an acute replication and then a set point. And so this acute phase is called primary HIV infections. Many of them are symptomatic, but there can be asymptomatic ones as well. 5 to 30 days after exposure, and a variety of symptoms which are nonspecific, and so you can't tell that you have an HIV infection. Very few of these might be hints, and this lasts for about 2 weeks. You may think you have the flu, and you really have an active HIV infection. Lymphoid cells, or lymphoid tissues, I should say, are routinely and actively destroyed throughout the body by virus replication. So this is a picture of the terminal ileum, the part of the small intestine near the large intestine. And normally, in a normal human, HIV negative person, the wall of the intestine is lined with collections of lymphoid cells. These are these yellow patches here. And you can see in an HIV positive person, after a few weeks, all those cells are gone. The virus has wiped out all of the collections of lymph cells, of T lymphocytes, CD4 positive cells, because the virus is replicating in them. So although this looks nice to you, nice and clean, it's not good. It should look like the one on the left. So here's the course of infection. You have virus introduced at time 0. You have a short acute phase, of up to 6 months or so. And during that phase, we have virus replication. So virus is on the bottom here, HIV RNA copies per mil of plasmid. So plasma, you can see a peak in virus replication. It goes down to a certain set point after 6 months or so, and then you last many, many years with that low level of RNA. Now on the top, if you look at the CD4 T cells, the number of T cells in the blood, you can see that's in red. There's an initial dip during the acute infection, then you recover, but then over the ensuing years, the numbers of cells go down slowly and slowly and slowly until you get into well, this is basically a persistent infection now. You get into a phase where you have too few CD4 cells to be able to respond to other infections, and then typically death is because of other opportunistic infections. And your CD8 cells are pretty much steady during this period of CD4 cell decline, but during the AIDS period, they go down as well. And you can see at the end, the titers of virus go way up because there's no more immune system to get rid of the virus at all. It's being destroyed. And while it was held in check here in the middle, it can no longer do so towards the end. One of many genes that controls susceptibility is the CCR5 gene, the gene encoding this co-receptor for the virus. And we know that people that have a deletion in this gene are protected against infection. There are a number of those people, 4 to 16% of individuals of European descent. So this deletion has, for some reason, been fixed in this population. We don't know why it's there. It may have been another pathogen many years ago. There have been many speculations. But I think I've told you the story of the Berlin patient, the German AIDS patient, who had cancer, and his physician was going to give him a bone marrow transplant to cure the cancer. And that involves ablating the bone marrow with radiation and then giving a donor's bone marrow, which will repopulate the bone marrow, get rid of all the cancer cells. And the doctors, and the guy had also at the same time AIDS. So the physician said, let me find a CCR5 delta donor. He did. And this is the only patient who's since been cured of AIDS. He has no more virus in him and he is healthy because his bone marrow was ablated. It was replaced with bone marrow donor from a CCR5 negative person. So this experience has led to the idea that we can take someone with AIDS, from which we can't excise the provirus at the moment, and take out their bone marrow or T cells, cut out the CCR5 gene with CRISPR now. It used to be with other nucleases, but now with CRISPR, and put that back into the person and make them HIV negative. So lots of people are working on this, either trying to cut out the provirus itself or cut out the receptor, because if you can populate a person with CD4 positive cells that don't have CCR5, they should be resistant to infection. So I think probably in the next 10 years or so, you'll see therapy based on this. Problem is getting the precursors of all your immune cells in your bone marrow, taking CCR5 away from them. That's not easy. You have to do a bone marrow ablation, which is associated with a lot of mortality. So if we can figure out how to do it without ablating the bone marrow, this would be very good. Now, during established infection, that long period where the virus is present, you have replication, major reservoirs outside of the blood, the gastrointestinal lymph sites, which I showed you, the central nervous system, the genital tract. You make a lot of virus each day, and the half-life is less than 6 hours and maybe 30 minutes. Incredible production of virus and turnover every day. And this, of course, leads to many, many, many mutant viruses. This shows you some of the compartments of HIV infection. Of course, in the blood, there are lymphocytes which are infected, generate lots of virus every day with a half-life of as little as 6 hours and 1.6 days for generation. So as soon as you make more CD4 positive lymphocytes, they come from the bone marrow into the blood, they're infected, and that generates lots and lots of HIV. But we also believe that there are other compartments of latently infected cells. And for some reason, when the virus gets into a progenitor, a hematopoietic progenitor, the cell that gives rise to all the immune cells, the provirus goes in and it becomes silenced. It doesn't make any more virus. And that's the latent reservoir that we have of HIV we can't get rid of yet, and which we are trying to get rid of. And again, when you take triple therapy, all this virus is gone. what's left is the provirus infecting these long-lived populations of memory cells or hematopoietic precursors. If you stop your drugs, they make virus again and you get infected. So that's the problem in dealing with AIDS. So we have a couple of different kinds of clinical presentations. The typical progressor I've described, you get infected, you have primary HIV infection, you have a long period of low-level virus production, then you progress to AIDS. There are people who are called rapid progressors. They're dead within a couple of months. This whole thing is accelerated. And then we have non-progressors, people who get infected. They have a primary HIV infection. They make very, very little virus for years, and they never progress to AIDS. And of course, we want to know what's with these people, because maybe we could take what we learn and help some other people as well. And we call these people elite HIV controllers. Again, they get infected. They maintain normal CD4 counts and undetectable viral loads. We know it's there. They have proviral DNA for sure. For many years in the absence of antiretroviral therapy. This is about 1 in 300 infected persons. And there are multiple determinants of this long-term ability to survive with infection. One of them is an MHC type called B57 and B27. And this influences the T cell responses to one of the viral proteins. These people do have persistent viremia, but it's very low. And these viruses are not attenuated. So it's not a virus thing that this person got an attenuated virus and it's not pathogenic. It is a host difference. And as I say, HLA is one of the main determinants. So just to remind you, the MHC, which sometimes they call HLA, the MHC proteins of the host cell will display peptides from viruses. So here's an infected cell. If it's HIV infected, the MHC is displaying a viral peptide, and a CTL here is detecting it as foreign. People with, now these genes that encode these MHC proteins are very polymorphic in the human population, and some people have proteins that seem to be better at presenting all the variable HIV peptides that are spun off during infection. So as the virus replicates in us and mutates, it's making proteins that are slightly different, and in a normal progressor, some of these proteins aren't displayed or aren't recognized by CTLs, so we get immune escape. But for some reason, the MHCs of these elite controllers are really good at presenting whatever mutant or altered viral peptide is displayed. And we're trying to use that to some advantage in the long term. Now, this virus just trashes the immune system. If you want to see what the ultimate immunosuppressive virus is, this is it. Now, it infects CD4 positive T cells, it kills them, and that trashes the helper ability, of course, of these cells, but it also reduces the number of CD8 T cells. It messes up B cells, they abnormally proliferate, and they don't make good antibodies any longer. It also results in abnormalities of dendritic cells, macrophages, NK cells. For example, it decreases the cytotoxic activity of NK cells. So it has a global immunosuppressive effect, really remarkable. So the consequence is when you get into a situation of having very few CD4 positive T cells, you begin to get opportunistic infections that you would normally be able to fight off, but since your immune system is trashed, you can't get rid of them. And these include protozoan infections, bacterial infections, fungal infections, and you remember pneumocystis was that first hint that something was going on in those 5 patients from LA. 5 people with pneumocystis, rarely seen, only in immunosuppressed people, and all of a sudden, and it's because the immunosuppressive ability of the virus. As you're infected, your immune system is responding. It's activating and trying to reduce infection. It turns out that HIV replicates even better in activated immune cells. So it's fostering its own propagation. You also get cancers associated with HIV infection, lymphomas by EBV, Kaposi's sarcoma, and also anogenital carcinomas. And finally, the virus causes neurological symptoms as well. Meningitis, myelopathies, neuropathies, and what's called the AIDS dementia complex. Let's see how some of these arise. So this is a nice summary of all the things that happen during the entire course of infection. We have our acute phase, that early acute infection where you can't really tell that you have AIDS. The virus establishes its reservoirs very early on. and you have continuous low virus production. Now, during this asymptomatic phase, you may have some symptoms sporadically, which include some of the indications here, none of which are terribly specific for AIDS, except perhaps thrush, leukoplakia, and shingles. And then finally, when your T cells get very low, you get all the opportunistic infections. If you had tuberculosis, it may often be activated. And then, as I said, you get a variety of microbiological infections, cancers, and neurological symptoms when the cells go below 200 CD4 plus per mil. The neurological symptoms arise because the virus enters the central nervous system and replicates in cells. So here on the top left is a capillary passing through the central nervous system, and viruses are coming from the capillary into the CNS, which is shown below. Virus can come within cells or as a naked virus. The virus can infect a variety of cells or get taken up into a variety of cells in the CNS, including microglia. So microglia are expanded on the right here, and when infected with HIV, they make a variety of cytokines, pro-inflammatory cytokines, chemokines, all part of the immune defense system. But in the CNS, these have neurotoxic properties directly on neurons or other cells as well. So all of the symptoms of neuro-AIDS, we think, is a consequence of the release of these immune mediators caused by infection at that site. HIV leads to increased incidence of cancers. 40% of people with HIV develop various cancers, which is an effect of not having an immune system, of course. Your immune system is important for scanning for cancer cells as they arise, but also you're producing high levels of cytokines. I just showed you all the cytokines made in the brain, but these are made everywhere. And one of the effects of cytokines is that they make cells divide. What happens when cells keep dividing and dividing? You know the answer, right? They accumulate mutations, they become transformed, they can become tumors. So making cytokines continuously, because you have this virus present in you for years and years, causes these tumors to occur. And if you have any viruses in you that have oncogenic potential, we all do, EBV, HHV8, HPV, those will cause cancers as well. Now, Kaposi's sarcoma was one of the early indicators of AIDS in the US. This is actually an old disease described in 1872, mainly in older immunosuppressed Mediterranean men. It's actually caused by herpes virus, HHV8, or Kaposi's sarcoma herpes virus. So these old men were getting herpes virus infections. The sarcoma can show as a skin lesion, as shown here, but you also get internal tumors as well, which you cannot see. And this happens in 20% of HIV-infected men, 2% of women and transfusion recipients. So again, it's in many people have HHV8. If you get immunosuppressed because of AIDS infection, HIV infection, you will get cancers as a result. Now, can we make a vaccine to prevent infection? We know that during infection, the viral load goes up, and then antiviral immunity kicks in and brings down the virus load to a certain set point. So we should, in theory, be able to do that. But some really puzzling questions. Why does the virus persist? Yet we have these antibodies in us during the course of this infection. And how does it end up killing all of the immune system at the end? One of the reasons is that HIV escapes from neutralizing antibodies. So let's say you're infected with this purple HIV, replicates in you. Within 2 weeks, you develop antibodies that will recognize it. By then, the virus has replicated so extensively. Remember, it's an RNA virus, it's making a lot of mutations. The virus has already changed its envelope like a protein. Now it's orange. And these antibodies you've made are not effective at blocking infection. They may make it worse for all we know. Okay, so your body responds and makes some orange antibodies, but now the virus changes again and has a blue coat. So that is now not neutralized by any of your antibodies. So this happens continuously in you. The virus is continuously evolving, and you are making antibodies throughout this whole period, but none of them do any good. So we need to make a vaccine that will somehow get around this. There have been a number of trials of the traditional vaccine method. One of them is a trial called RV44, which was done in Thailand, where there are lots of sex workers, a very high at-risk population for AIDS, So you have to find a country with a high-risk population so you can assess the effectiveness of your vaccine. And this was a prime boost regimen. They first injected people with a pox virus of birds, a canary pox virus that encoded the envelope, gag, and pall proteins of HIV. And then it was followed by an injection of a recombinant GP120, the glycoprotein of the virus. So 2 shots. This was put into 16,000 volunteers in Thailand. 6 prime and 6 boost injections. 12 injections. Can you imagine the logistics of this clinical trial? Plus, you have to take all these 16,000 people, and half of them, of course, get placebo. Half of them get the real vaccine. And you have to tell them, do not have unprotected sex because you'll get AIDS. You have to tell them what you don't want them to do, but they will do it anyway, and that's why you can test your vaccine. But you have ethical responsibility to counsel them not to do what you would actually like them to do. Now, this particular vaccine lowered the rate of infection by 31% compared with the placebo. But these are the numbers. Out of these 16,000 volunteers, there were 74 infections in the placebo group and 51 in the vaccine group. Only 100 infections of these 16,000 people. Of course, they lost some along the way, but this is not very good. 30% is just not good enough. So we still have a lot of work to do. One of the cool things that's happened in the last 5 years or so is people have found broadly neutralizing antibodies against the glycoprotein. So here's the glycoprotein of HIV. There would be the viral membrane at the top. And these simply indicate different sites where monoclonal antibodies in different colors are binding to the glycoprotein. About 20% of infected people have these. You can purify them from people, and they will neutralize all of the subtypes that arise. These are very potent neutralizing antibodies. They recognize conserved epitope. People are trying to figure out how to use these in some kind of a vaccine. And one of the approaches is really neat. This has been done by David Baltimore at Caltech. He took the gene encoding one of these broadly neutralizing antibodies, okay? And he put it, here it is, IgG, heavy chain, light chain. So remember, antibodies have 2 chains. You have to put both chains in and produce both of them in a cell to get the antibody. So he cloned the heavy and light chains of one of these broadly neutralizing HIV antibodies into a adeno-associated virus vector. Remember, adeno-associated virus, single-stranded DNA genome with 2 T's at either end, inverted terminal repeats. You can make recombinant DNAs, and you can make virus particles that will now deliver the antibody gene to a host. So he tested this out in a humanized mouse. So you take mice and you replace their immune system with immune components from humans, because otherwise the virus doesn't grow in mice. And he immunized mice with this construct. Now again, this is making antibodies. And these mice make tons of antibodies. And here is the result on the top. This is the number of HIV copies per mil of mouse plasma. So the immunized groups are all the red bars. So they're completely protected from challenge infection. And the mice that receive the vector all get infected. And these mice make like milligrams of antibody per mil. And at the bottom is a summary of this. So the control mice, percent uninfected. The control mice that don't get the antibody, it goes down to 0 within 18 weeks. And these are multiply challenged mice. And at the top, the red bar, these animals who get the vector with the antibody never get infected. So this is very, very stunning. Of course, it's in mice. And now this is going to go into people, phase 1 and phase 2. So the idea would be, if you're a high-risk population, you could get this. The problem that I see, of course, you're going to make antibody for the rest of your life. These AAV vectors are persistent. I don't know what kind of effect that would have, but for certain populations, it would certainly be one way to go. Now, another way is shown here, and this is using a macaque model, an SIV, and making a vector out of a simian cytomegalovirus. You know, we have human cytomegaloviruses that persistently infect us for years. And their simian counterparts. So they took a simian CMV and it cloned into it the glycoprotein of SIV. So this is a pure SIV model. And then they vaccinate these animals and challenge them with SIV. So the blue on the y-axis is the viral load in the plasma. We're looking at SIV. This is time post-infection in weeks. Now all the blue lines are animals that got the vector. Vector only, CMV vector, no SIV insert. You can see they all develop a viremia and they're not protected as you would expect. The other animals, they're all in different colors here on the right, and they're shown down at the bottom. These are animals that got the SIV glycoprotein in the simian CNV vector. You can see many of these are protected. There are a few animals that develop bursts of viremia here and there, but again, this is considered to be another promising vaccine. Now here we're delivering a viral antigen, but the key is that these CMV vectors are persistent, just like the adeno-associated virus vector. So again, one or a few shots of this, the virus could persist in you for life and give you lifelong protection. So we'll see what happens with these two approaches as they move into people. This is really what we're working with. We start in 1921. A chimp with SIV in Cameroon gets captured by a hunter. That hunter becomes patient 0. And over the next years, this virus causes 78 million infections and 39 million deaths. I find this remarkable that a virus can go from one monkey to so many people, spread silently for so many years. But this is really what we're faced with on Earth, with so many animals that have different viruses. Who knows when the next big one is going to be coming? That's why I find this story so remarkable.